Welcome to the training session on Phenotypic Drug Susceptibility Testing for the BPaL regimen using the MGIT system without TB-Exist software. This training session was prepared by the International Tuberculosis Research Center. This training material is designed to provide a technical overview of pDST for the BPaL regimen within the LEAF-TB-OR project. Most scientists working on LEAF-TB-OR projects are presumed to be well-trained and have a significant amount of experience. Given this, our training will not go into detail on the routine steps of these experimental protocols, but instead will focus on the critical aspects of the protocol that may impact test performance, accuracy, and results. Two methods commonly used to evaluate the susceptibility of MTB to new drugs are the MGIT system and the microdilution method. This training is mainly focused on the MGIT method, which can be standardized and utilized in all 7 countries evaluating the BPaL regimen. Although the use of MGIT is recommended in the LEAF-TB project, supplemental material for the microdilution method can be obtained from the ITRC upon request. Please keep in mind that this training will focus on the use of MGIT for pDST without the use of TB-Exist software, which can be used in the Epicenter data management system with the MGIT. This is because not all participating sites have access to the TB-Exist software. If you have a TB-Exist, you can simply follow the manufacturer's instructions regarding the tube settings and obtaining results. Before we start the experiment, I would like to remind you of a few important things. First, equipment validation is the most important step before starting an experiment. Please ensure that your equipment is validated within the period stated in the management plan. If the validation expiration date is close and the patient enrollment period is not fully covered by the current validation certificate, I strongly recommend that you validate the equipment before starting the experiments. Second, you do not need to be afraid of starting this new experiment. I understand that all participating countries have experience with betaquilene pDST using the MGIT system. If you have completed this, you will note that pDST for linezolid and pratomanid is not that different from pDST for betaquilene. The main difference is the drug substrate being evaluated. The principles, procedures, and other details are nearly identical. Finally, you need to focus on how to perform these experiments accurately. How many samples you can handle in a day, or how fast you perform the experiments might not matter at all. Just stick to the basics. Pipette slowly and accurately, and use a new micropipette tip every time you pipette. This slide is designed to remind you of the principles behind the MGIT system and how they apply to MGIT-based pDST for the drugs in the BPaL regimen. Principle 1. A known concentration of a test drug is added to one of the tubes. Principle 2. The growth in the tube is compared with that in the growth control tube. Principle 3. If the test drug is active against a strain, it will inhibit the growth. Principle 4. The drug will reduce the fluorescence relative to that of the growth control, which will display increasing fluorescence because growth is not inhibited. Principle 5. Growth is monitored by the instrument, which automatically interprets the results as either S or R. As most of you already know from your experience with the betaquilene and linezolid evaluations, WHO provides the critical concentration for determining resistance to betaquilene and linezolid. You may refer to the WHO recommendations from WHO Technical Report on critical concentrations for drug susceptibility testing of a medicine used in the treatment of drug-resistant tuberculosis in 2018. However, because pretomanides was recently introduced, not enough data have been collected to set CC for pretomanide resistance. Therefore, the minimal inhibitory concentration method is still being used for pretomanide. The LIFT-TB project will use the MIC of pretomanide and the CC of bedaquiline and linezolid to monitor drug resistance using MGIT. As described in slide 5, 1 microgram per milliliter of bedaquiline and linezolid will be used as a CC for MGIT pDST, while a series of concentrations of pretomanide will be tested to determine the MIC in each tested sample. The concentration range for pretomanide was provided by the TB Alliance and was determined based on the data from the previous NICS-TB study. This material does not cover determination of pDST of clinical isolates toward pretomanide using the MIC assay. However, after you assess pretomanide pDST using the reference MTB strain H37RV, you can begin testing clinical isolates. You can follow the procedure described in the following slides to obtain reliable pDST results for pretomanide using the MIC assay for the patient samples. The MIC of pretomanide for H37RV should be between 0.06 and 0.5 milligram per milliliter. If your MIC is within this range, you can consider your experimental results valid. The overall process is presented on slide 6. Before you start, you need to prepare the followings: the working solutions for each drug from the original drug stock solution, MGIT tubes, and inoculum. Then, inoculate the isolates into the MGIT tubes. After incubation, you can read the results and produce a record within the report. Slides 7 to 10 show how to prepare the drug stock solutions. 1. For each drug, dispense 10 ml of DMSO into a 15 ml tube or equivalent using a 10 ml pipette. 2. Weigh 40 mg of drug powder using a high-precision electronic scale. This step is critical. Any errors in the measurement can affect the results. 3. Add the 40 mg of powder to the 10 ml of DMSO prepared in step 1. 4. Vortex the mixture thoroughly until the drug particles and sediments are completely dissolved. This will produce a 4 mg per milliliter stock solution of each drug. 5. Aliquot 100 microliter of each stock solution into sterile screw-capped vials using a 200 microliter micropipette. 6. Once reconstituted, the stock solution vials should be transferred to separate cryo boxes for each drug and stored at -20 degrees Celsius for up to 6 months. 7. Once thawed, use immediately and discard any unused portion. Working solutions of each drug should be prepared immediately before each experiment. Remove the stock solution of each drug from the stock boxes, thaw at room temperature, and keep on ice before use. Do not use leftover working solutions again, but discard after each test. To prepare the drug working solutions, you need to complete 2 different dilution processes, one for bedaquiline and linezolid, and another for pretomanide. This is necessary because critical concentration will be used as the test criteria for bedaquiline and linezolid, while the MIC will be used for pretomanide as explained in slide 4. A. Preparing working solutions of pretomanide. Several more dilution steps are required to make 7 1:2 serially diluted working solution named WS1 to WS7. 1. Prepare DMSO to dilute pretomamide stock solution as follows. A. Dispense 958 microliter of DMSO into a 1.5 milliliter screw cap tube using a micropipette and a 1000 microliter micropipette tip. This will be used to produce WS1. B. Dispense 800 microliter of DMSO into 6 2 milliliter screw cap tubes using a micropipette and a 1000 microliter micropipette tip and label these tubes WS2 to WS7. 2. Prepare WS1. Transfer 42 microliter of the pretomamide stock solutions using a 100 microliter or 200 microliter micropipette tip to WS1 from step 1A. Mix by pipetting thoroughly and vortexing briefly. 3. Prepare WS2 to WS7. Transfer 800 microliter of WS1 using a 1000 microliter micropipette tip to WS2 from step 1B. Mix by pipetting thoroughly or vortexing briefly. Repeat this process to prepare WS2 to WS7. B. Preparing working solutions of bedaquiline and linezolid. 1. Dispense 979 microliter of DMSO into 2 1.5 milliliter screw cap tubes using a micropipette and 1000 microliter pipette tips, one for bedaquiline and the other for linezolid. 2. Transfer 21 microliter of the appropriate stock solution using a 100 microliter or 200 microliter micropipette tip into the DMSO prepared in step 1. 3. Mix by pipetting thoroughly or vortexing briefly. 4. This will produce a working solutions of each drug with a final concentration of 84 microgram per milliliter. The final concentration in the MGIT tube will be 1 microgram per milliliter. The test MGIT tubes should be prepared and organized as follows. 1. For bedaquiline and linezolid, the MGIT tubes should be placed in the 5-tube AST carrier. A. Label one tube as the growth control for bedaquiline and another for linezolid. B. Add 0.8 milliliter of OADC to each tube using a 1000 microliter micropipette tip. C. Transfer 100 microliter of each working solution using a 100 or 200 microliter micropipette tip to the appropriately labeled MGIT tube. D. Place the tubes in the correct sequence in the appropriately sized AST set carrier and then put the MGIT tubes without drugs and OADC into the remaining carrier positions. 2. For pretomamide, use the 8-tube AST carrier. A. Label one tube GC and the remaining tubes WS1 through WS7. B. Add 0.8 milliliter of OADC to each tube using a 1000 microliter micropipette tip. C. Transfer 100 microliter of WS1 using a 100 or 200 microliter micropipette tip to the WS1 MGIT tube. Repeat this process for WS2 to WS7. D. Place the tubes in the correct sequence in the appropriately sized AST set carrier. Inoculum can be prepared using either MGIT culture or solid culture. To prepare the growth control, please see step 3 at the end of this section. 1. Preparation of inoculum from a positive MGIT tube. The first day that the instrument detects a positive MGIT tube is considered day 0. For the preparation of test inocula, if the tube shows positive culture results after day 1 or day 2, no dilution is required. However, if the tube shows positive culture results after day 3 to day 5, it cannot be used directly as an inoculum, but instead must be cultured again using a midget culture tube. 2. Preparation of inoculum from a solid media. A. Using a 5 ml pipette, add 4 ml of distilled water to a 15 ml conical tube containing 8 to 10 glass beads. 2. Scrape as many colonies as possible from the growth using a sterile loop, trying not to take any of the medium and transfer it to the conical tubes containing glass beads prepared in step A, and resuspend the bacteria. C. Vortex the suspension for 3 minutes to break up the larger clumps. D. Let the suspension sit for 20 minutes without disturbing. E. Transfer the supernatant to another conical tube and let it sit for another 15 minutes. F. Transfer the supernatant, which should be smooth and free of clumps, to a third conical tube. G. Adjust the suspension to a 0.5 McFarland standard. Do not adjust below a 0.5 McFarland standard. H. Dilute 1 ml of the adjusted suspension in 4 ml of distilled water. Use this diluted culture for the BPaL drug testing tubes. 3. Growth control inoculum. Transfer 0.1 ml of the prepared inoculum from either a positive midget tube or solid medium into 10 ml of distilled water to prepare the 1 to 10 GC inoculum. 1. Inoculation. A. Inoculate 500 microliter of the inoculum suspension prepared as described in slide 10 using a 1000 microliter micropipette tip into each tube except for the positive and negative control tubes. B. Inoculate 500 microliter of 1 to 100 diluted GC inoculum suspension from step 3 on slide 10 into the GC tubes using a 1000 microliter micropipette tip. C. Mix thoroughly by gently inverting them 3 to 4 times. 2. Check for contamination. To ensure that the inocula prepared for either positive midget tubes or diluted from solid media used to set up the BPaL test is not contaminated, spread 0.1 milliliter of each suspension onto a blood agar plate and incubate at 37 degrees Celsius for 48 hours. Then, evaluate each plate for bacterial growth. If there is no growth, then the BPaL test can proceed. If the BAP shows growth, discard the BPaL test tubes and repeat the inocula preparation. Set the tubes in each AST carrier and follow the midget user's manual for DST instructions. Everything after this step is the same as the procedures used for routine evaluations. The reporting form presented on this slide is developed by TB Alliance. The complete form can be downloaded from the same webpage of the ITRC where you find this training material. This brings us to the end of training session for Phenotypic Drug Susceptibility Testing for the BPaL OR project. We hope you have enjoyed this session and found this material useful. If you have any questions or inquiries, please contact the ITRC at mlab.itrc@gmail.com. Thank you very much.